Okay, hello you guys. In this video, we will be looking at the last topic of the 2.2 notes. And in this last topic, topic 4, we're going to be looking at something called active transport. And so let's just jump right into it. So active transport, this is when, we're still talking about the movement of substances across the cell membrane into and out of the cell. Just like in passive transport. But the big difference here that you guys are going to see is that this requires energy now. So now we're going to talk about the movement of molecules across the cell membrane in a way that is going to demand energy. And usually that energy is going to come from ATP and that's being made inside the cell. And so there's going to be an input of energy that needs to be met in order for this movement to take place. And so this is usually because in active transport, what we're going to do is we're going to move substances against their concentration gradient, not along their concentration gradient. So in passive transport, substances were moving down their concentration gradient from high concentrated areas to low concentrated areas, which gets it closer to equilibrium. But in active transport, we're going to look at solutes who are actually moving against their gradient, meaning that they're going from areas of low solute concentration, and those solutes are moving to areas that already have a higher solute concentration, which is actually going to get this thing further away from equilibrium, not towards equilibrium. By moving, we're going to make the side that's already more concentrated even more concentrated. And that is not something the universe wants. And getting further away from equilibrium, the universe doesn't like that. And so to do something the universe doesn't want to happen, you have to put in some energy to do that. So that's exactly what's going to happen in active transport. This movement is going to require specific proteins that are going to have to help move these things against their gradient. So this will for sure require proteins. And those proteins will be carrier proteins and not channel proteins. So if you guys remember, there's two types of transport proteins. There's channel proteins, which just have like a channel down the middle that things can freely flow through in and out of the cell. That only works for passive transport when things are moving in the direction that the universe wants them to move. So like going down their concentration gradient from high to low. But in active transport, this will for sure require carrier proteins always, because you're going to need a protein to actually interact with that substance, like bind to it and literally push it to the other side. And so that's what we're going to see here. So this picture is a good example. You see on one side, there's a higher concentration of hydrogen ions. A lot of times we call these protons, hydrogen ions. In science, or in biology, we call them protons. What's happening in this picture is the protons, the hydrogen ions, are being moved to the outside of the cell where there's already a higher concentration of hydrogen ions. And you can see that ATP is going to be required to do that. So this is called a proton pump. Anytime you hear the word pump, that's usually, almost always referring to active transport. It's literally pumping something into a place that it doesn't want to be. And that's what's going on in that picture. And so another good example of active transport that you definitely need to know for this class is the sodium-potassium pump. This is like a double example of active transport of substances moving against their concentration gradient. And so it's a great example, and it's also a very common protein pump that's found in living organisms. And there's a lot of sodium-potassium pumps in lots of different cells in your body. And they're actually really important for your muscular and nervous system, which we don't get into that much in this class. If you're taking AP Psych, you will definitely talk about the sodium-potassium pump when you talk about the nervous system and neurons, or anatomy and physiology. You'll learn about the sodium-potassium pump as well. But basically what's happening here, if I just walk you through this picture, the sodium-potassium pump is moving two different substances. It's moving sodium ions and potassium ions. Now, specifically, it's moving sodium ions out of the cell and moving potassium ions into the cell. And in both of those situations, those ions are moving against their gradient. So sodium is going to move out of the cell where there's actually already a higher concentration of sodium, and potassium is going to move into the cell where there's already a higher concentration of potassium. So it's doing active transport times two. And for two different substances, they're going against their gradient. And this does require ATP to do this. And then if you guys look, there's a specific number that you guys need to know. So specifically in the sodium-potassium pump, there's 3 sodium ions being pumped out of the cell for every 2 potassium ions being pumped back into the cell. And so remember those numbers. And this will be important in a second too, but 3 sodium ions out of the cell for every 2 potassium ions that go into the cell. And so that the fact that it's pumping these ions in a different proportion is going to create something called an electrochemical gradient. And an electrochemical gradient, what that is when you have first of all, a chemical gradient, which a chemical gradient is just saying that there's a different concentration of chemicals on both sides of the membrane. Like I said, there's more potassium inside the cell, there's less potassium outside the cell, there's more sodium outside the cell and less sodium inside the cell. So that's what we call a chemical gradient. But this also creates an electrical gradient. An electrical gradient is when there's a difference in charge across the membrane. So on one side of the membrane, there's a different overall charge than on the other side, which is going to make one side more positive and one side more negative because the electrical charge doesn't match. And so the sodium-potassium pump is creating not only a chemical gradient, but an electrical gradient. And the reason why there's a chemical gradient being produced is because of the whole 3-2 ratio that I was talking about. Sodium and potassium ions both have a positive 1 charge. So sodium has a positive 1 charge, calcium has a positive 1 charge. And since this guy is always moving 3 sodiums out of the cell, each with a positive charge, but into the cell it's only moving 2 potassiums, which both have a positive charge, what's going to happen is when you have a cell who has literally thousands of these sodium-potassium pumps doing this every moment of every day, you're always going to have more positive charges outside the cell than inside the cell. So this guy is unfairly dispersing the charges. He's pumping out 3 positive charges outside, but only 2 positive charges inside the cell. And for that reason, the outside is going to end up with a more positive charge than the inside of the cell. So that creates an electrical gradient. So we would say that the outside is more positive than the inside, which then means that the inside is negative compared to the outside because it's not as positive as the outside, which that kind of confuses students because you guys don't really see negative charges here. But if one side is less positive than the other side, well then, and relatively speaking, you are negative compared to that other side. Or just think of it as less positive, however you want to think of it. But that's what's going on here. That is all summarized here, and it tells you where there's a high and low concentration of sodium, potassium, and which direction these things are going. And so that electrical gradient that I was just talking about, that difference in charge, can be measured by in terms of something called a membrane potential. A membrane potential is a measurement of the charge difference across a cell membrane, and it's measured in millivolts. And so whenever you have a separation of charge, when 2 sides of a membrane have a different charge, that creates a voltage. Like that's literally the definition of a voltage, is when there's a charge difference, a separation of things that are differently charged. And that's exactly what's going on in here with the sodium-potassium pump embedded in these cells. There's going to be a difference in charge, and we can measure that. And when we measure that difference in charge, that's what we call the membrane potential. Now so for example, neurons, your guys' neurons in your nerve cells and even the neurons in your brain, they have like thousands and thousands of sodium-potassium pumps in each nerve cell. So every nerve cell has just a bunch of these sodium-potassium pumps on their cell membranes. And so because of that, the membrane potential is measured as -60 to -70 in a resting nerve cell. And the reason it's negative is because we're measuring the charge of the inside of the cell relative to the outside of the cell. And like I said earlier, the inside of the cell is more negative or less positive than outside the cell. So that registers as a negative voltage. And that's all because of the sodium-potassium pump helping to establish that electrical gradient. But anyway, that's active transport because it requires ATP, and we're moving substances against their concentration gradient. This is not something the universe wants. This is getting the thing to be further away from equilibrium, not closer to equilibrium. And so that's the big idea here. Another example of active transport is something called a co-transporter. This is an interesting example as well. A co-transporter is a protein, a transport protein that's actually moving two different substances across the cell membrane. Now, what's going to happen in this situation is one of the substances that it's moving is going to be moving down its concentration gradient from high to low, which is what the universe wants. So one of the substances that it's moving is going to go down its concentration gradient. But the other substance that it's moving is going to be going against its concentration gradient. So from an area that has a lower concentration to an area that has a higher concentration, which is not what the universe wants. And so in a co-transporter, you have both things happening at the same time. So one substance is being moved against its gradient while one substance is moving along its gradient. Now in this picture, there's a good example of something called the sucrose proton co-transporter. This is found in plant cells. And this guy moves both sucrose and hydrogen ions across the plant cell membrane. And what's happening here is the hydrogen ions are moving down their concentration gradient from high to low, which is what the universe would want. But the sucrose that's being moved is being moved against its gradient. It's being moved into the plant cell where there's already a lot of sucrose. There's already a lot of sucrose inside the plant cell, and the plant wants to move even more sucrose into the plant cell. And so that's what's happening here. So the sucrose is being moved against its gradient. Hydrogen ions are being moved along their gradient. And so that is an example of active transport because we do have a substance moving against its gradient, and the movement of this substance, the sucrose in this case, does require energy. But in this case, the energy doesn't actually come directly from ATP. So this is a good example of energy being required, but not coming directly from ATP within the cell. The energy that this guy is using is actually the movement of these hydrogen ions. That movement of hydrogen ions down their gradient is what's providing the energy. That's actually energy, that kinetic energy, that movement, that flow of hydrogen ions, just like water down a river. That's energy that can be harnessed. And this protein is pretty cool. It can harness that energy, the energy from these hydrogen ions moving down their gradient, to power the movement of sucrose against its gradient, which does require energy, which comes from these hydrogen ions moving down their gradient. That's what's happening in a co-transporter. Now, ultimately, really, if you zoom out, look at the big picture here, it really does tie back to ATP eventually, because to maintain this concentration of hydrogen ions outside the cell, where there's a higher concentration of hydrogen ions outside the cell wanting to come into the cell, there's a proton pump, like I showed you on the first slide, that's pumping them constantly out of the cell against their gradient, which does use ATP. So technically ATP is being used, just not directly by this protein. This protein doesn't touch ATP, doesn't use ATP, but it is doing active transport, not directly using the energy from ATP, but using the energy from these hydrogen ions going down their concentration gradient. So that's what a co-transporter is. This is just a specific example. But whenever two substances are moving, and one of them is going down its concentration gradient, one of them is going against its concentration gradient, that is a co-transporter, which is active transport, because one substance is going against its gradient. And then the last thing in these slides is talking about the movement of large particles across the cell membrane, or a large amount of substances across the cell membrane all at once. We call this bulk transport, which is when you have a large amount of something going into or out of the cell all at once. This could be a large macromolecule, maybe like a protein, which is really big, or a large carbohydrate or something. And to get that across the cell membrane, you can't go through a protein. Like a protein is not big enough to move this giant molecule into or out of the cell. So there's these processes that we're going to look at in a second called endocytosis and exocytosis that involve these vesicles forming and fusing with the cell membrane. And so we'll look at each of these in a second. What I want to emphasize right now, though, is that this is active transport, because the formation of these vesicles that we're about to look at and the movement of these vesicles inside the cell to the cell membrane, it does require a lot of ATP, but the pictures never show you all the ATPs that's being used. So whenever you have these large vesicles being carried towards the cell membrane, or the cell membrane engulfing something and forming a vesicle, this will make sense in a second, but whenever any of that's happening, endocytosis or exocytosis, you need to know that there's a lot of ATP involved. It's not just happening by magic. And so, but in the pictures, you'll never really see the ATP. And so just kind of be aware of that. So let's start with exocytosis. In exocytosis, what's happening is there's a vesicle inside the cell that's carrying some stuff, and that stuff needs to be secreted from the cell or released from the cell. So that vesicle comes and fuses with the cell membrane, and then by fusing with the cell membrane, is able to then release and secrete that material that it was carrying. So maybe these are like enzymes, digestive enzymes in your stomach cells that are making these guys by the rough ER and the Golgi, modifying them and packaging them into this vesicle, and here they're being carried to the cell membrane and then being released out of the cell. That process is called exocytosis. And so that's what's happening there. And this does require ATP, this vesicle moving to the cell membrane and fusing with the cell membrane. There's a lot of ATP involved with the cytoskeleton and motor proteins, all of which we don't really go into detail in this class, but just take my word that there's a lot of energy to do this. So this is why it counts as active transport. Now, the opposite of that is endocytosis, and this is when something outside the cell is being engulfed by the cell via the formation of a vesicle that now forms inside the cell. So the plasma membrane of the cell surrounds this object outside the cell and then forms this vesicle that pinches into the cell, and now it's inside the cell. Which again, this requires a lot of ATP. They're not showing you, but the cytoskeleton that's allowing this kind of movement to take place is using a lot of ATP. There's two types of endocytosis. Well, I guess I talk about three types of endocytosis in these notes. One of them is called phagocytosis. Phagocytosis is when it's a large solid molecule being swallowed by the cell. So again, this could be like a large macromolecule, like a large protein or a large carbohydrate that's huge that the cell wants to then take into the cell. Sometimes it could be a whole nother cell, like your guys' white blood cells. Your white blood cells find bacteria cells and swallow them just like this via phagocytosis. And then another version is called pinocytosis. Pinocytosis is when it's a solution that the cell is engulfing that has a solute dissolved in it. So there's some kind of chemical dissolved out here in this solution that the cell is just taking a drink of. I think it literally translates to cell drinking, pinocytosis, whereas phagocytosis is cell eating. And so it's taking in this solution by surrounding the plasma membrane, forming this pinching inward of this vesicle, just like in this example or this picture here. And then the last type that I put on here is something called receptor-mediated endocytosis. And this is when there's actually this is like a more specific version of endocytosis where there's proteins on the cell membrane who are waiting to bind to a very specific molecule out here that's binding to this specific receptor. And once it starts binding to those receptors, it initiates this endocytosis process, which now takes in those molecules. So it's kind of like a specific targeted thing that the cell is looking for that's going to wait for it to bind to these receptor proteins. But that's, I think, it. Just know that endocytosis and exocytosis, those are active transport. There's a lot of ATP not being shown in this picture being used to form these vesicles and this movement that's taking place. But this last slide just gives you a nice little summary, a very short description of each of these types of transports that we've talked about in topic 2 and topic 3 of these notes. Well, topic 2 and topic 4 of these notes. And these different examples of passive transport that don't require energy and these examples of active transport that do require energy. But that's it for these notes. We're officially done. You guys can work on answering these questions, and we'll talk about some of this in class. That's it. Thank you, guys.